Good morning, everyone. It is with a great joy and enthusiasm that we welcome you to the second edition of GBAI Day. I'm Marcela Mendes, professor of the School of Agronomy of the Federal University of Goiás, UFG. On behalf of Professor Rafael Tassinari and the entire organizing committee, I would like to warmly greet all attendees. The first edition of GBAI Day, held entirely in Portuguese language last year, featured lectures by experts in GBAI interaction, including Professor João Batista Duarte and Alexandre Coelho from University of Goiás, Caio Olímpio from the University of Viçosa, Gustavo Marcati from University of São João del Rei, and Dr. Luiz from Embrapa. That edition brought together almost 500 participants from 12 countries. As with the first edition, our speakers this year have been carefully selected. This morning, we will hear from Dr. Hans-Peter Piepho of the University of Hohenheim, Germany, Dr. Larissa Vendrame from Embrapa, Brazil, Dr. Daniela Bustos-Korts from the Austral University of Chile. In the afternoon, we will be joined by Dr. Fabiani Rocha from Corteva Agriscience, Brazil, and Dr. Diego Jarquin from the University of Florida, United States. Each speaker will be introduced in more details prior to their section. I'm pleased to share a brief overview of this second edition. We have reached over 1,000 participants from 71 countries, formed primarily by students and industry professionals. Most participants work with annual crops. As many of you know, GBAI interaction is not merely a statistical concept. It lies on the heart of some of the most pressing challenges in modern plant breeding. Understanding these interactions is essential for selecting cultivars that are not only productive, but also resilient and stable, especially in the face of climate change and the increasing global demand for food quality and yield. The decision to host this event online, free of charge and entirely in English, was not only practical, it was also strategic. It allows us to broaden the reach of the knowledge being shared here today. Democratizing knowledge begins with making it truly accessible to those who seek it. The free registration reaffirms our commitment to academic inclusion, opening this space for exchange and learning not just to established specialists, but also to young researchers and students. Beyond technical updates, this event is a declaration of our dedication to open, collaborative, and accessible science. Science that aims for real impact, built through networks and shaped collectively. This event is organized by LAMP, Precision Breeding Lab, coordinated by Professor Rafael Tassinari, who is also the driving force behind this initiative, and by the study group EuGeM, composed by graduate and undergraduate students affiliated with the grad program of Genetics and Plant Breeding of UFG. We thank our partner studies groups from the Intergen network and the startup TheCrop for supporting the promotion of this event. I will now share a short video introducing our graduate program.
The graduate program in genetics and plant breeding is based at the School of Agronomy of the Federal University of Goiás, located in Goiânia, the capital of the state of Goiás in Brazil. Goiânia lies in the heart of the country and is known for its quality of life, urban infrastructure, and culture. It is part of Brazil's Central West region, a key player in national agricultural production and a hub for agribusiness development. Our program offers master's and doctoral degrees aimed at training professionals to lead in teaching, research, and innovation. The faculty includes professors from UFG's School of Agronomy, Institute of Biological Sciences, and Embrapa. Students benefit from modern laboratories, experimental fields, advanced technologies, and collaborations at both national and international levels. Our research lines are structured around three main areas: plant genetics and genomics, aiming to explore the genetic basis of traits using molecular biology, genomics, and bioinformatics. It also includes studies on beneficial microorganisms that support sustainable agriculture and tissue culture.
The laboratory also works with the in vitro propagation of sugarcane varieties, including the production of transgenic plants focused on the introduction of genes related to tolerance to herbicides and biotic stress. Another line of work under development is the micropropagation of cannabis sativa genetic materials, with the aim of improve the levels of cannabinoids, especially CBD and THC, through clonal cultivation and control of cultivation conditions.
Breeding of cultivated species, focusing on crop improvement to develop high-performing genotypes, using both classical and modern breeding techniques. These efforts integrate high-throughput phenotyping technologies, quantitative genetics approaches, and the study of G/E interactions.
At the intersection of quantitative genetics, geoprocessing, GIS, environmental data and file experimentation, we focus on environmics, using high-throughput environtype covariates, mainly from satellite sensors, to better understand G/E interaction and improve prediction accuracy. We apply mixed models and statistical tools to analyze multiple environmental trials, combining genomic selection, phenotypic data, and programming, mainly in R and Python. This support precision breeding in the context of modern agriculture.
Research is primarily conducted on major crops such as maize, common bean, rice, eucalyptus, pepper, cotton, soybean, and sugarcane.
The sugarcane breeding program at UFG is a research and extension group dedicated to developing sugarcane varieties adapted to the growing conditions of central Brazil. Our team includes geneticists, plant breeders, plant pathologists, field and lab technicians. We are so proud to be a part of a network of 10 Brazilian universities working closely with one another and with the sugarcane industry. Varieties developed through this program currently account for over 65% of all sugarcane cultivated in the country.
Conservation and breeding of native plants from Cerrado. Research in this area emphasizes the conservation and utilization of both native and cultivated plant species. These research efforts led to the establishment of in vivo germplasm collections of native fruit species.
For over 30 years, the Federal University of Goiás has studied fruit trees native to the Brazilian Cerrado, such as piqui, baru, and cashew. Our research contributes to the domestication, genetic improvement, and conservation of these species. We also study genetic diversity to promote sustainable use and identify markers that help these species adapt to climate change. To strengthen these efforts, we created the Combaru Network, which focuses on conservation, genetic improvement, and the first reference genome for Baru.
Our curriculum includes core and advanced courses in molecular genetics, statistics, plant breeding, biotechnology, bioinformatics, quantitative genetics, genetic resources, and scientific writing. Emphasis is placed on biometrics across courses and research. This structure builds a solid theoretical and practical foundation aligned with each student's research focus. Beyond formal coursework, the program promotes academic development through initiatives like EuGeM, the student-led study group in genetics, conservation, and plant breeding. EuGeM organizes regular meetings, seminars, and workshops that connect theory with practice and foster student leadership. It also hosts SimGeM, the annual international symposium on genetics and plant breeding. We are committed to internationalization, offering opportunities for international research partnerships, academic exchanges, and internships. We encourage an inclusive academic environment grounded in sustainability and innovation. If you're looking for academic excellence, engagement with cutting-edge research networks, and training in genetics and plant breeding, the Graduate Program in Genetics and Plant Breeding of UFG is the right place for you. Connect with us on social media.
I encourage you all of you to visit our website and follow us on social media to learn more about the work carried out by our team. We'll now begin the lecture by Dr. Piepho. Dr. Hans-Peter Piepho is a professor of biostatistics at the Institute of Crop Science at the University of Hohenheim in Germany. He has PhD in plant breeding at the University of Kiel with focus on yield stability. Since then, he has made substantial contributions to the field of linear mixed models, statistical analysis of agronomic traits, and quantitative genetics. He's recognized as one of the most cited researchers in these areas. Dr. Piepho, it's a great honor to have you with us today. Please feel free to begin your presentation.
Okay, I can go? Okay. Hello, everyone. My name is Hans-Peter Piepho. It's great to be here and give the first talk in this conference. Great to know there's quite a crowd out there. So what I'm going to do is going to be based on this paper here, which was published last year, and it provides a review. And the perspective that I'm taking is that I'm a farmer, and we have this big trial network in a multi-environment trial, which represents the target population of environments, and I'm in there somewhere. The trial network is represented by trial locations. Those are the blue circles. And then the blue dot you can regard as the mean in the target population of environments. So here's the plan for my talk. And just before I start, I will say that, well, number one, I've got quite a few slides, so I won't be showing all of them. You may see me skipping a few at some point. I may also not be getting all the way to part 3, but let's see. My talk is going to be primarily about models, and there's very little in the way of actual empirical results, so be prepared for that. Right, and with that, I'm ready to go. So I want to talk about analysis of variance models first, really going back to basics. Then part 2, we'll talk about Findlay-Wilkinson regression, its history, and how this gradually transitioned into what's now known as factor-analytic models. And then part 3 extends this type of modeling so that we can include observable environmental covariates. So to start part 1, I want to go back to some very basic and seminal papers that are 90 years old almost. Or some of them, this one is 91 years old, and just read you some citations from quotations from those papers. So, and this is about really very basic assumptions about the types of model. They're all going to be mixed models that we're fitting. And the key message here is that environment should be a random factor for the purpose that I'm focusing on, which is making predictions, making recommendations for a farm, for a location in the target population of environments. So this is from Immer in 1934, and he says, insofar as these 6 stations constitute a random sample of conditions to be found over the entire state, and that these 2 years are a random sample of weather conditions to be encountered in future years, general recommendations may be drawn up for the entire state with reasonable assurance that the variety or variety recommended will prove to be consistently superior in most places of the state and in most years. Here's another seminal paper, Yates and Cochrane, who basically invented what's now known as Findlay-Wilkinson regression. And they basically say the same thing. So let me read it out to you, just because this is such an important point. Any experimental program which is instituted to assess the value of any particular treatment or practice, or to determine the optimal amount of such treatment, should therefore be so designed that it is capable of furnishing an accurate and unbiased estimate of the average response to this treatment in the various combinations of circumstances in which the treatment will subsequently be applied. That's your target population of environments. The simplest and indeed the only certain way of ensuring that this condition shall be fulfilled is to choose fields on which the experiments are to be conducted by random selection from all fields which are to be covered by the subsequent recommendations. They do go on to qualify that it is usually impossible to secure a set of sites selected entirely at random, but an attempt can be made to see that the sites actually used are representative. But averages of the responses from such a collection of sites cannot be accepted with the same certainty as would be the averages of a random collection. So they're basically saying, yes, in terms of what we are trying to achieve, environment should be a random sample. In practice, it may not be so easy to get a strictly random sample, but we should try to get as close as possible and then model our results accordingly. So talking about modeling and analyzing these data, we're going to be using mixed models, and then a key choice is for each of the factors involved, whether it's regarded as fixed or random. And once the status of each factor as either fixed or random has been established, one may use the convention that if an effect involves at least one random factor, it should be modeled as random. So if environment is a random factor, it follows automatically that variety by environment interactions are to be modeled as random as well, regardless of whether variety is modeled as fixed or random, by the way. Emma has this recommendation, which basically says we're going to be comparing variety means, and we're going to be testing varieties, and we're going to be using mean squares for the interaction of variety by environment as our error term. And Fisher basically makes the same point. So he says, in fact, if our concern is to ascertain not merely the best variety on the aggregate of the fields actually used, but to ascertain which is the best over the whole area deemed suitable for this type of crop within the region from which the sites of the experiment have been selected, the comparison between varieties and interactions of varieties and places will be the more appropriate. He's referring to the F-test here. For if the sites have been chosen at random from this area, a significant difference in this comparison would indicate at the level of significance used varietal differences applicable to the whole area. And Yates and Cochrane basically make the same kind of statements. Okay, so this is about ANOVA models. They were talking primarily about ANOVA models. Yates and Cochrane were also talking about Findlay-Wilkinson regression, to which I'll be coming in part 2. For now, ANOVA model. So you can either have a two-way model with genotypes and environments, or you factor the environments by locations and years, which I think is good practice. And then we're dealing with a three-way ANOVA model. This is just your three-way ANOVA model. And then depending on your choices regarding the factors, effects may be decided to be fixed or random. So in my development here, genotype will be fixed. I'll very briefly talk about genotype random later. But the key point is that environments are random, and so locations are random, and years are random as well. Now, to compare different options as far as the use of a fitted model is concerned, we can look at differences between two varieties. This simplifies a lot of things, primarily because effects not indexed by variety, such as location main effect, year main effect, and the location by year interaction drop out. So you look at the difference of two varieties in a randomly picked location and year. You can then ask, what's the expected value of the variety difference in the whole TPE? So you take the expected value over this difference in equation 3. And it turns out that the difference just depends on the difference of the two main effects for the two genotypes in question. And assuming that all the random effects have zero mean constant variance and are independent, as we usually do in simple ANOVA type models, we can conclude that the probability that in a randomly picked environment, variety I is better than variety H, this is going to be the case whenever the difference between the two, alpha I minus alpha H, in terms of the variety main effects, is larger than 0. This is very simple. And this was pointed out by a number of authors, and one very nice paper which has a risk perspective, farmer's perspective really, is Eskridge and Mum cited here. Okay, so this is the rationale actually for recommending a variety with the largest mean across all the environments in the whole TPE. And this is perfectly fine from a breeder's perspective, because you're breeding as a breeder for the whole TPE, and you just want to be best on average. That's the best you can hope for, really. But now I'm a farmer, so how about my farm? How can I use the trialing network to get the best information for my farm, the best recommendation for my farm? Things are a bit more complicated. So in the best case scenario, I'm able as a farmer to identify the trial location that is actually very close to mine, or actually identical to mine. If I'm lucky, the trial was actually, or the trials were actually conducted at my farm. So that's really the best case scenario. So in that case, I can actually condition on the location, which is my farm, ideally, and then ask, okay, what's the expected difference between two varieties? And that difference now depends on the two main effects for the two varieties, but also on the two variety or genotype location interactions at this particular location. And then we can make the same kind of argument that the probability of variety A outperforming variety H in a random year really only depends on on this contrast, which involves main effects and interactions of genotype by location. So what you would do is just take the estimate of the mean of each variety at that particular location across years and just make a comparison and pick the variety with a larger mean. Very simple. But there's a caveat, and that's the farmer's location is not necessarily one of the trial locations. So what do we do? And how, if I'm a farmer, do I actually best estimate what's going on at my farm based on the data that's available in the trial network, which does not include my location? Okay. I'm actually a little less ambitious here. I'm still looking at the best case scenario. I'll come back to that more ambitious task of, going to have to extrapolate from the trial network, which does not include my location. I'm going to assume still that one of the trial locations is the farmer's location. So we can then work out the variances of our estimate. So option 1, I'm using the long-term mean at the jth location. The variance of a difference, the difference of those two varieties that I'm looking at, depends on genotype by year interaction variance and the three-way interaction variance. There's another option. I can use the mean in the whole target population of environments, and that's actually going to be my main conclusion here, that for a farmer it's actually better to look at the mean in the whole TPE oftentimes. We can look at the variance of a difference of the mean of those two varieties in the target population of environments. What this does is it brings in the genotype by location interaction variance into the picture, but at the same time, that variance gets divided by the number of locations in the trial network, which may be quite large. So that addition to the variances may not be a big deal. But more importantly, the three-way variance gets divided not only by the number of years, but also by the number of locations, which is good. This is actually the main reason why taking the TPE mean, the mean of the varieties in the TPE, may be preferable. But we need to be a bit careful because, of course, the mean in the target population is also not the same as the farmer's location or the particular location we're looking at. So there's going to be a bias between the mean in the target population and environment or location J. And that's written down here. It depends on the two interactions of the two varieties for that particular location. Now, if we can assume that the location is a random draw from the TPE, we can work out the expected value of the squared bias. And we can then add that on and compute the mean squared error of a difference, which now includes this bias term. So to work out the trade-off between the two options, option 1 is I'm just looking at the means at that particular location J, versus I'm looking at the means in the target population of environments. To study that, we need to compare those two equations. And we can then work out the conditions under which option 2 is better. And the simple message is if the three-way interaction is large, then it's typically better to use the mean in the TPE, that is option 2. Okay. So we've made a heroic assumption so far, and that's the farmer's location is the jth location in the trial network, and we're able to identify that location. But often that's not the case. Now, the worst case scenario in that respect is we have to regard the jth location that we might consider picking and the farmer's location as two random draws from the same TPE. And then your mean squared error of a difference all of a sudden has 4 times the genotype by location interaction variance added. Okay, so this makes it worse in terms of option number 1. Okay, we can be a bit more optimistic and hope that the farmer is able, or we're the farmer, and we're able to identify the location in the trial network that is closest to ours in a particular sense. What I've done here, and then this factor of 4 in front of the genotype by location interaction variance on the right-hand side of the equation has this factor C instead of 4, okay? And that factor C, we can determine that by simulation, which I've done using a simple simulation. I generated interactions from a normal distribution, and then I basically looked at the smallest value of the difference between the two differences. for those interaction effects for two varieties at my farm, which is going to be j equals 0, and the farm and the location in the trial network that I deem most similar to my farm. So I pick the smallest among all the locations that are in my simulator data set, and then I compute in the simulation what is the MACD. And it turns out that this factor C, it drops quickly from 4 down to, and it can drop down to 0. It does so fairly quickly. That's good, but there's yet another caveat, and we must acknowledge that identifying the most similar trial location may not be so easy for a farmer, especially when there's a large number of locations. And the immediate idea to pick the location that is closest in terms of distance may actually not behave quite differently and less similar than other locations in the network. So that's not easy. We've also assumed that there's only a single trait to look at, but we're always looking at multiple traits. And we've also assumed that we're only looking at a single pair of varieties. But in actuality, we need to look at many pairs to pick the best. So all of this means that what we have in this equation for the mean squared error of a difference is really just a lower bound for what can be realized in practice. And the main problem is really summarized here. If I'm a farmer, so I'm the red circle, then I'm, in terms of a single variety, I'm 2 interaction effects away from any location, any individual location I might pick from the trial network. Whereas I'm only 1 interaction effect away, and that's the green arrow here. I'm only 1 interaction effect away from the TPE mean. And that is, in a nutshell, why it's usually better to rely on the TPE mean for the varieties, rather than trying to pick the location that I think is most similar to mine. Okay. I did not talk about year effects here. In terms of the mean squared error of a difference, we need to add on in terms involving interactions with years, but the same terms need to be added for the 2 options, so that doesn't change the relative merits. So there's an important general conclusion here. For a farmer, the estimated variety mean in the TPE is likely to provide a more reliable basis for variety recommendations than the variety mean at the trial location considered to be most similar. And I can judge the relative merits of the TPE means versus location means via the mean squared error of a difference in my framework here, because environments are modeled as random. This is really important. That's one of the main reasons why I would like to model locations as random in these kinds of exercises. By comparison, if I choose to model environment as fixed, I get these equations down here, and basically I can still estimate a genotype mean, all right, but there's a big difference, which may actually not show in this equation, but everything I do is conditional on a specific set of environments that were in my trial network. Everything is conditional, and I cannot really extrapolate to the whole TPE. Well, I could just take that mean and say I'm going to use it, but I cannot attach a standard error or a mean squared error or anything, any measure of uncertainty to it based on the fitted model. Now, I can do cross-validation, so there's other ways of getting some measure of uncertainty. But if I want to do things inside the mixed model framework, then I can make no progress at all at this point if I want to make a recommendation to a farm with respect to a farm that's not in the trial network. Right, now here's a set of slides that I will be skipping. This is just to point out that the same results basically hold when genotype is modeled as a random effect, which I may want to do for a number of reasons. I want to do genomic prediction. I want to borrow strength across agroecological zones. The equations just look a bit more complicated because I'm using BLUP, but the message is the same, and the relative merits actually tend to be the same as well. So I'll skip those and move on directly to part 2 on Finlay-Wilkinson regression and how these gradually evolved into what is now known as factor-analytic models. And you will see the same issues of very basic questions of, is environment fixed or random? Is genotype fixed or random? Come back in my slides. So you all know this thing here, Finlay-Wilkinson regression. This is was originally proposed by Yates and Cochrane. I recently came across two papers that are actually a bit older than Yates and Cochrane, 1938. So it's really fun to go back in history and really go back to the roots and see where all of this is coming from. So this is Finlay-Wilkinson regression, where you regress yield of the individual genotypes in the individual environments against the environmental mean, where the environmental mean is computed from the varieties, from the yields of the varieties tested in those environments. And the average slope of that regression is 1. And then we can judge whether we have a high input or a low input variety. So if the slope is larger than 1, we have a high input variety that's very responsive to favorable environments, and a low input variety if the slope is smaller than 1. And people have used this to characterize what's denoted as stability, phenotypic stability, terms like resilience and so forth all go with this. Right, so what's the model? It's simple straight line regression. It's just that now the regression is indexed by genotype and environment, because we have those two factors involved. To keep it simple, I'm not factoring environments by years and locations for now, but it's understood that that's what you should be doing if you have multi-year data. Right, so we have intercept and slope to characterize each genotype, and then the slope is what we're interested in, because that characterizes the sensitivity. This equation assumes that we have an observable covariate for the jth environment. Right. Do we have that? Well, what we do in Finlay-Wilkinson regression, as I said, we plug in the genotype mean, or sorry, the mean for the environment. So the mean across all the genotypes in environment J, simple arithmetic mean. That's Finlay-Wilkinson regression. That's all fine and splendid, and been used by a lot of people and still is, and it's pretty good. The one thing that's not so nice is that we actually use the response y on both sides of the equation. We use it as our predictor variable on the right-hand side, but it's also the response on our left-hand side. I mean, it says on the left-hand side, I have this Greek letter eta, which is really the expected value of the ith genotype in the jth environment. So to get a full model for the response, I would need to add on the plot error or the field error, whatever. But the fact is that if I want to fit this model, I need to use the response on the left-hand side as well. So using the response on both sides is not a good idea from a statistical point of view. And it's been criticized by a lot of people. And, you know, just try to imagine you want to use this model and you want to simulate data from this model. I wouldn't know how to do that because I want to simulate the response. Okay, if I want to simulate the response, I need actually the responses in my hands already, so I can plug them in on the right-hand side, so it's not working. So what have people suggested is replace that mean by a latent effect so that they have at least a good model. You can say that this model underlies Finlay-Wilkinson regression and that the arithmetic mean of our genotypes in environment J is our estimate of this latent effect W. Now, if environment is a random factor, now we're moving to the random effects here, we can then say, okay, this latent effect is a random variable because it's indexed by environment and it has a normal distribution with some unknown variance. That's fine. We can go a step further and go to this slide, which looks almost the same, but there's a little difference, okay? So on this slide, I'm saying the average of my slopes is 1, as is usually the case in Finlay-Wilkinson regression. Whereas on the next slide, I've replaced the betas with lambdas. Very subtle change here, just a different Greek letter. And I'm imposing a constraint not on the slopes, but on the W's. I'm saying they have a unit variance. So saying I have a unit variance is a bit like saying the average of my slopes is 1. Not exactly, but it's comparable in some sense. So this is really the random effects version of Finlay-Wilkinson regression when environment is a random factor. And the context in which this type of model has been originally proposed is stability analysis, where really we are trying to characterize how does my variety respond in a bunch of environments, where really, again, conceptually, environment needs to be a random factor. Now, we can use this set of assumptions on the previous slide and work out what is the variance-covariance matrix of our vector of expected responses in a given environment J for all the genotypes. And then we have the variance-covariance matrix shown here, which has the slopes involved, and it has the variance of the deviations, which also need to be modeled. I haven't said how they're going to be modeled, but we can impose models there as well. So this is a variance-covariance structure. That's observation number one. Observation number two, this is actually recognized as a factor-analytic variance-covariance structure of order 1, a special case, just one set of lambdas. You can easily extend that to have multiple sets of lambdas. So this was originally proposed in a mixed modeling framework in the context of repeated measures by Jendrik and Schluchter. And Bev Gogol in 1995 was actually the first to propose this type of model as a random effects version of Findlay-Wilkinson regression. She did not use the term factor-analytic, but she was the first really to have a full REML framework for estimating this type of model, which is really great. You can assume regarding the deviations from regression that each genotype has its own variance. So you can say this is a stability statistic, and that's Eberhard-Russell regression, if you've heard about that. So that's just another stability measure that people have used. So in many ways, you can regard the choice of what's the best stability statistic as the choice of what's the best variance-covariance structure to describe my data in my multi-environment trial. Okay. So how can we use this model for prediction? I said my perspective is I'm a farmer, and now I want to have a prediction recommendation for my farm. We can't really make much progress here because for a new environment, this random effect W is not known. It's just another random draw from a normal distribution. So it doesn't change. So we can still just use the expected mean difference in the TPE. And we just have a different variance-covariance structure compared to the ANOVA model, or the variance-covariance structure that the ANOVA model induces. It induces what's known as a compound symmetry variance-covariance matrix, if you want a name, as opposed to the factor-analytic model you see here. So it's just that whole variance-covariance matrix may be quite big. So the uncertainty in those predictions doesn't go away. It's just been modeled a bit more sophisticated, and that's all. So to underline that somewhat depressing fact, I have this quotation, which may be funny for those of you who know The Hitchhiker's Guide to the Galaxy. If you don't, I recommend you read this. It doesn't have anything on plant breeding, but a lot about why the future is largely unpredictable. Okay, so have you got an answer? No, but I have a different name for the problem. It's just really reflecting what I just said. We have a different way of modeling the covariance, but we haven't really gotten rid of that uncertainty. Okay, I'll come back to that point, but maybe I can actually say, or should be saying right now, that the best way to reduce the uncertainty is use observable environmental covariance. Okay, and that's where a lot is going on these days. A lot of people are working on that, and I'm sure you're going to hear a lot about it today in the talks that come after me, which is good. Especially because that's going to be in my part 3, which I may not be able to complete. So a few variations to the Findlay-Wilkinson regression idea, or factor analytics. So what if instead of having an intercept for the variety, we have an intercept for the environment? And this may sometimes be a pretty good model. So here's an example where I found this useful on heading dates. The trait is heading date in perennial ryegrass. And I have a trial. I've used a trial, and this is really good. Not going to be my only real results. Sorry about that. So 113 genotype, 7 years. So years is environments, and that's a random factor. Okay, and heading dates a number of days after April. So here's the fitted model. So I fitted a regression line for each year now. Okay, just go back to the equation. You see there's still this multiplicative term lambda I times WJ, but now actually the WJs, they're random because environments are still random in my outline here. But now I'm not going to assume, which is usual in mixed models, that the random effects have zero mean. That's not a sensible assumption here. We want to assume a non-zero mean, because these are the slopes, and you can see very clearly here that the average of the slopes is nowhere near zero. Okay, so we can't assume expected value of the slopes is 0. Now, what's interesting here is that if you take the topmost regression line, that's for a year, that's a late year. Okay, so flowering is late. So all the flowering dates of the different genotypes are actually pretty compressed. Whereas this one here, for example, which has a much steeper slope, is an early year where some of the genotypes start flowering pretty early, and then you have just varieties just starting to flower step by step, and it's just spread out across a much larger span of time. So that's a pretty good fitting model for flowering times and for heading dates. And here are the fitted slopes and intercepts. So this is basically reverse Finlay-Wilkinson, if you will, but insisting that environment is still a random factor and variety is fixed. So it's interesting to look at the variance-covariance structure this implies. Looks a bit different, and it has this factor-analytic touch as well, but it really is different. And the main difference really is that these lambdas now all of a sudden appear both in the variance-covariance structure, but not only there, they also appear in the mean. Why? Because the expected value of my slopes is 1. That's a constraint I'm opposing, but the main point is that it's not 0. Okay, so the lambdas do not. They also feature in the expected value, and the variance-covariance structure becomes a quadratic function of the mean, which is interesting too. And here's a plot of the sample variances against the sample mean for my varieties, and you see this downward drop, and a quadratic model gives a pretty good fit. Okay, this is also again pretty opposite of what you usually see. You usually see the variance increasing with the mean. Here it's actually dropping. But the reason is that, as I said, if you've got a late year, things get compressed, and if you've got an early year, things get spread out. Okay. Now, how do you fit this model? I haven't actually said how I would usually fit the model, but I'm always using REML. I'm not knowledgeable enough to do very fancy Bayesian things, although I'm keen to do that, and it's a great framework. But if I use REML, I can't use that here. Why? Because my lambdas, which are in the covariance structure, also are on the means, and the whole idea of REML is to kick out the fixed effects and then fit the residual, or use the residual likelihood for those residuals to estimate the variance parameters. Now, if I kick out the fixed effects, if I kick out the means, then I've kicked out the lambda, so I have no information left to fully estimate my variance covariance structure. So REML doesn't work. I can use maximum likelihood, and I've used that a couple of years ago, quite a few years ago, actually, I realize, 25. But I don't think anybody has used this model after my publications, sadly. And maybe it's because of this fact. This is actually more tedious to fit. But if you're a Bayesian, and I'm pretty sure there's a lot of Bayesian people out there, this is a piece of cake. And it may be a good model to fit, actually. Okay. Now, if we add on both main effects, both for genotype and environment, and we still assume genotype fixed, environment random, then all of a sudden it's much easier, because now I can assume that the average of the W's have a mean of 0, that the expectation is 0. Why? Well, you could assume otherwise, but then any non-zero expectation would actually be absorbed by the main effect alpha I. So imagine plugging in some expectation for W in my equation 21 here. Let's say a 1. Then you've got lambda I times 1. So alpha I plus lambda I would be confounded, or would be aliased, whichever one you want to call it. So you can drop one of the two in the expectation. So this works fine. So this can be estimated by REML, no problem. Now, a more dramatic change, actually, because now we're assuming that genotypes are random and environments are fixed. So I'm making that same swap that I did at the end of my ANOVA slides. And I should pause for a second, because this is the type of model that is appearing in the vast majority of publications that use factor-analytic model, which is quite different compared to what I've shown you so far. And what I've shown you so far is the history of the whole thing. Okay, So environment fixed, genotypes random. So my W's now become random. And I have fixed intercepts, which I dubbed eta. So I'm using Greek letters for fixed effects here. And I have a fixed slope for the environment. This is my reversed Finlay-Wilkinson regression, but now also swapping the fixed and random assumptions. So this type of model has the same inferential limitations that the ANOVA model has if you model environment as fixed and genotype as random. Okay, well, most importantly, it's really the fixed environment assumption that comes in here. Okay, it's difficult to extrapolate to other environments that are not in the trial network. So people have come up with suggestions. How do you even define a main effect or a mean? People have suggested what's called the generalized main effect. So this generalized main effect involves the WI, which is a genotype-specific term, okay, but also the average of the lambdas. We need to average across the lambdas if we want an average expectation. So I've just averaged the model across the locations. The main point is that this average is now again conditional on the set of environments in my trial network. So can't extrapolate and can't assess the uncertainty of predictions outside of that network, outside of that set of environments, at least not based on the fit of my mixed model, I should say. I said you can do cross-validation, okay. You can always do that. But you can't do it in a mixed model framework, and that's something that I do not like so much. You can model genotype, you can add a random genotype main effect, and then you can compare genotype means across the specific set of environments. Something very important to keep in mind here, although you now have a main effect, you might think I can just use the main effect to make recommendations. No, you can't. You need to keep in mind the W's, the slopes. So it's really the random effect A, the random intercept, plus the random slope W times the average of the lambdas that needs to be used and considered to work out the genotype mean in the TPE. And then you can look at the difference of the two. Okay, same inferential limitation as before. Okay, now what do you do if both environments are and genotypes are random? That's nasty, at least for a poor frequentist like myself. A piece of cake if you're a Bayesian, I suppose. There's some iterative methods out there that people have proposed that you can use, or you can use a two-stage approach. But this would need to be explored. I haven't really looked very closely into this. But that's a very interesting approach, of course. You know, you want random environments for the reasons that I hope I've made plausible, but you also want random genotypes because you have marker information and stuff. So what do? Well, you want to do something like this. Okay. And what I've shown you involved just a single latent environmental factor. Of course, you can extend this to multiple such factors, and there's a lot of papers out there that do this. And you want to factor by years and locations, and there's some papers out there that have done this. Right, but the most important step now is this one, where, as I've indicated before, if all of a sudden we have an observable environmental covariates, we can then start thinking about, can we, using those, to model the latent variable W. And here's just a straight multiple linear regression on the observable covariates. The important point is it's observable covariates. So if I have, for a new location, I have all the covariates, I can just plug them in, I can predict W, and I can make very specific predictions potentially. So that's great. There's one big caveat, and so this is really changing. That's potentially a game changer, but there's one big caveat that I want to point out, and I'm actually getting quite close, I guess, to the end. Maybe 2-3 more minutes. So this is all nice, but the one thing we need to keep in mind, there's different types of covariates, and unfortunately, not all of them for a given location are constant over years. The most interesting ones, or many of the interesting ones, are varying from year to year. Anything related to weather, of course, varies from year to year. And I want a prediction now because I'm planting, and I would need to have a prediction of what the weather conditions are going to be at the critical stage of grain filling or flowering in the upcoming season. And unless we have very accurate weather forecasts that have, a range of a couple of months forward, that's going to be a very uncertain thing. So we have a lot of uncertainty in those covariates. And that, I think, is sometimes overlooked. So I don't want to end on a too pessimistic note here, but it needs to be really kept in mind that a lot of the covariates are time varying, and we need to deal with that uncertainty. What I really think is we actually need a model for the covariates. We need another mixed model, not just for the for our yields or phenotypic responses. We need another mixed model to describe the variability of our covariates across years. And then maybe combine the model for yield and the model for the covariates to come up with an overall model, including the facility to assess the uncertainty. So people have used environmental covariates in recent years. There's a lot of hype there, and you'll hear more about this, I'm sure, in the other talks. And people use cross-validation, and we do it all the time as well. And what do? We leave out, we say we do leave one out, cross-validation. So we leave one environment out is a good idea. So that mimics the situation that a farmer is in. So it's a new year, it's a new location. If I'm in extension service, I don't know what exactly is happening at that farm. So I'm leaving out the location. What does that mean? I'm leaving out the yield data, okay. But then virtually always people just plug in the covariates. And the covariates may involve a lot of time varying covariates. And so that's not really the situation the farmer is facing, where he doesn't know what the rain is going to be during grain filling. So he's dealing with a whole distribution of values that the covariate can take. So that adds to the uncertainty and can bring you depressingly close to the very simple prediction, which is just your genotype mean in the TPE. But there is potential, there is room for improvement. There is hope that bringing in covariates in this way can really, it's not going to be a quantum leap, I don't think, but it's going to be a little extra information. Just as when Marcus came along, people started doing QTL, and there was a lot of hype, and now everybody uses it, but it's not really just completely changing the picture. There's still a lot of groundwork to be done by the breeder day to day, as had to be done 50 years ago, and I see the same thing happening here. I just want to put that into perspective. So regression models, we can do regression models with environmental covariates, and there's a bunch of variations to it. And since I'm going to have to omit part 3, I can say that environmental kinship is one of the variations to this regression idea, and Diego Jarquin will be the last speaker today, and I'm sure he will talk about that approach in his talk, so that's good. And Daniela is on the call as well. Her talk may actually touch on the other approach that people have used, is to use the covariates to stratify the environments into what are known as environmental types, typical scenarios of how the weather might evolve over the growing season, and then cluster the environments and then use that clustering to come up with a more sophisticated model for G/E. And that type of model can also be used for making more targeted, more specific recommendations. Right, so here's a summary. Environments should be modeled as random. So this way we get an inference for the TPE. Genotypes can be fixed or random. Those are both options, but environments should be modeled as random, is my view. The most basic model is a 3-way ANOVA model. Finley-Wilkinson regression can be regarded as a regression on a latent environmental variable. And if that variable is random, then this induces a factor-analytic variance-covariance structure. And there are several variations. And when environments are modeled as fixed, then FA models suffer from the same inferential limitations as simple ANOVA. And the way forward is to model W, this latent variable for the environments, using observable covariates. And on that note, I should probably end.
Thank you, Dr. Piepho. Are you hearing me well?
Yes, very well.
Hi. Thank you for the excellent presentation. We had around 180 viewers during your presentation on YouTube. We received some questions. If you have some time, can I?
Sure, sure.
Thank you. So we received the question of Winifred Amongi. Thank you for the very informative presentation. I'm wondering what informs choice a GBAI model. For example, how do I choose among compound symmetry or Finlay-Wilkinson diagonal main plus diagonal, etc.?
Okay, yeah, very good question. I didn't talk about that. So if you use REML, then what people usually do is they use AIC, the Yakaiga information criterion, which is computed from the residual log likelihood, and it involves a penalty for the number of parameters. So there's a penalty for model complexity. So what AIC tries to do is it tries to strike the balance between model realism, which means very complex models. We have a lot of flexibility. So the most flexible covariance structure is unstructured, actually. But that has so many parameters, you can usually not hope to be able to estimate that model. So you want a more parsimonious model so that your data can actually support the model. But if it's too simplistic, you don't want that either. So you want to strike the right balance. And AIC does that. If you want to use AIC, you should be using, you can use REML, but if you use REML, you need to be careful and make sure that your fixed effects structure remains the same as you vary the covariance structure. And the reason is that the likelihood is really the residual likelihood, which is based on the residuals. And you can only compare likelihoods between different models if the so-called data is exactly the same. And if you use REML, your data is the residuals. And the residuals depend on your fixed effects. So if you want to also select on the fixed effects and you want to use AIC, well, think twice, maybe use F-tests, your world type F-tests to do that. If you want to use AIC to select on fixed effects, you need to switch to maximum likelihood, full maximum likelihood, so that your likelihood comprises the fixed effects. Now, there's some suggestions in the literature. You can also still use REML. So there's a very nice paper by Ari Verbela and a package that he wrote, but I think you need to have AS-REML. Is it called AS-REML Plus? So what he suggests is you can basically, you want to use REML because we know REML estimates of variance components are less biased than maximum likelihood. So that's why everybody uses REML. So use REML to estimate the variance components, but then plug those into the full likelihood and then compute AIC. That to me is really what he suggests. There's a recent paper in Euphytica in 2023 where this is described. No, sorry, it's a 2019 paper in the Australian Journal of Australian and New Zealand Journal of Statistics. Yeah, that's my REML answer. If you're a Bayesian, you use other techniques, some of which are also based on information criteria. Yeah.
Thank you. We have another question from Caio Olimpio. Great talk, Professor Piepho. To what extent are on-farm and on-station trials comparable in the context of GBAI evaluation and variety recommendation?
Oh, I love that question. I love that question. I mean, on-farm is so great from a farmer's perspective, for sure. I think they should just be used side by side if you have access. If I'm a farmer, then I would probably rather believe in on-farm trial data than in on-station. On-station is artificial compared to my farm because it's smaller plots and everything very controlled. But you can use both side by side and maybe go and extend the model. So if you have both on-farm and you have on-station data, small plot replicated, on-farm, typically unreplicated, much larger plots. So a lot of people think, oh, it's not replicated, it's not even a real trial, is it? Well, if you have this multi-environment trial perspective and the TPE perspective, every additional environment you can get hold of is very valuable because you're sampling more G by E. So every additional farm, even though it's unreplicated and even though it cannot test all the varieties, it's usually going to be just a subset, it's going to be extremely valuable and can all be integrated. We've had one experience where we had a direct comparison between on-station and on-trial, on-farm data. This was canola, and it really was on-farm trials, no unreplicated big plots as I described. And what we did is we fitted an extended model where we have 2 genetic effects for each variety, one under on-farm conditions, one under on-station. And we modeled it as random, and then we fitted a genetic correlation between the two. And it turned out the genetic correlation was really high. We also looked at heritability on farm and heritability on station, and the heritabilities were quite comparable. In other words, the precision was actually quite comparable between the two. Opposite of what a lot of people would have thought. So on-farm is extremely valuable. And I don't mind unreplicated, actually. And, you know, there's some big breeding companies that do a lot of unreplicated trials for the precise reason that they want to sample a large number of environments. So rather than having a few replicated trials with 2 or 3 reps, I would rather have a few more locations and go unreplicated. Yes, it's less accurate with respect to the individual trial, but as I've tried to explain, you know, an individual trial, although it's really right next door, is not as informative usually as a mean across several such environments. And I think this is also true when it comes to using environmental covariates. We need a lot of environments, actually. I didn't point that out. You know, you do a multiple linear regression on the environmental covariates. If you pick a single variety, what you do is just a simple multiple linear regression. Okay, and the samples in your data is really just the environment. So if I have 10 environments, the maximum number of covariates I can fit in a multiple regression exercise is actually 9. Okay, but I don't want to exploit those 9 degrees of freedom fully, so I would better just do 3 or so I have enough any degrees of freedom left to estimate the variance structure. We have tons of covariates, so we need tons of environments, really. So it's actually even more important to have a lot of environments. There are ways to simplify the model, and you know, Jarquin had this great idea to use the covariates to do the kinship approach, which in my view is an approximation to an unstructured variance covariance matrix for the intercept and all the slopes we need to fit for an individual genotype. So that's an approximation. I have a feeling that it's a very, very rough approximation. Okay, so use lots of environments. So use on-farm if you can. Sorry for a rambling answer here.
That's great, thank you. I think we have a little more time. Frederico Mandabula asked, what are the advantages and limitations of modeling environments as random effects compared to fixed effects in G-by interaction analysis?
Yeah, great question. I forgot to point out what I think is the main. There's several reasons why people use a model where environments are fixed and genotypes are random. I think one big reason is we have lots of genotypes and a small number of environments. So fitting Finlay-Wilkinson regression or any factor-analytic covariance structure with environments fixed and genotypes random is quite easy using REML in that setting. Lots of genotypes, very few environments. Another reason why people like this kind of model is especially if you have very diverse environments, you may actually find scale differences. Like you look at the spread of your genotypes, you know, they may all align in a very similar rank order between different environments. Think of the heading date example that I showed you. But then in some environments, they're pretty much spread out. And in others, they're more compressed because there's a lot of stress, so everything goes down, but maybe the ranking is still the same. So such scale differences can be modeled using lambdas. That's where people have used this type of model. But in those settings, I mean, I'm in Germany and in Central Europe, we don't have such big diversity in environmental conditions. So we hardly ever use factor-analytic models here, to be honest, because simple ANOVA models actually work quite okay. And generally, for the reasons that I've tried to explain in my talk, I would actually rather go with a simpler ANOVA model than fitting a more sophisticated model, which I think is more realistic, but then have this inferential problems. There's another part to the answer that I wanted to give. Well, if you turn it around, it's just more complicated. So if I started out looking into these models because I was interested in stability. So the reverse perspective, random environments, fixed genotypes is fine if I want to estimate the stability, assess the stability for a few genotypes. But I need lots of environments to actually sample the G by E and get a reliable estimate of stability. The simplest stability statistic of all is just the variance of my genotypes yields across my environments. Okay, and then if you look at basic results of the sampling variation of your sample variance as an estimator of that variance, it's huge. And you can work out, if you want to measure that variance down to a small coefficient of variation as a measure of precision, like 10%, for example, you need 200 environments, which is huge. So that's the big difficulty with any stability assessment, at least in terms of those basic mixed models that I showed you, where you don't have environmental covariate information. Things get better, I think, potentially when you have environmental covariate information. Then you can, estimate slopes, and potentially you can estimate those a bit more reliably. And more importantly, you can actually use those to actually make predictions for environments where you have the covariate information as well, with the caveat that there's time varying covariates which cause you trouble. Yeah.
Thank you. Do our time, I think I will send the last question. We had a few more, but it's from Renato Gomide de Souza. In scenarios with high interannual discontinuity, how stable are the aging structure and genotype loadings in FA models? And to what extent does this affect the extrapolation to future years?
Yeah, another very good question. How stable? I mean, your question, I hear that it's implying you're thinking in terms of, I have a single year of data. I have data this year. I'm fitting this model. And then I want to use that model, and then I want to make a prediction into the next year. That's difficult because you are essentially, even in the simple ANOVA model, yeah, you can fit a model that says, I have a genotype main effect, I have a location main effect, because that's all that varies in a given year. But your genotype main effect confounds the actual year genotype main effect plus the genotype by year interaction. And you have the same problem with Finley-Wilkinson or any factor-analytic model. So you're estimating all of those parameters. You may be estimating genotype-specific effects. You may be estimating location-specific loadings and so forth. But they're all confounding location with location year effects and genotype with genotype year effects. So you've got this instability. I think what's necessary to deal with that is actually fit a three-way model, which involves the factor year. So you need multi-year data. Now, in a routine breeding application, you don't usually have that. You really have to take fast decisions. So you want to take current year's data, get a result out, and be done with it. But I think there's a lot of potential in keeping, we have all this data, breeders have all this data that they've collected for years. Make use of that data. Use your long-term data. When there's time during the year, build a three-way model, estimate the variance components, and then use them. We've done something like that where we actually said, okay, we're going to take the breeder's perspective, we're taking this year's data, but we're only using it to estimate the blobs. We're using the long-term data to come up with estimates of the variance components, and then we can actually separate G and G by Y effects, for example. So we get more realistic blobs. Our predictions are not going to be much more accurate that way, but at least we're accounting for the three-way structure that always is there, even if we have a single year's data. So at least we can come up with a realistic assessment of uncertainty, which we can never have if we only use a single year's data, this year's data, to do everything, estimate the model, make predictions and everything. The uncertainty is really just the uncertainty for the estimation for what happened this year. This is a retrospective exercise, which doesn't really help for making predictions into the future. So we really need three-way data, we need multi-year data, and we need to have the year factor explicitly in the model. And we have it in the covariates anyway, so we need to have that kind of modeling for the covariates, as I've indicated, as well. Okay.
Dr. Piepho, on behalf of the organizing committee, I would like to extend our sincere thanks to you for this insightful presentation. We are grateful for the time and effort you dedicated to sharing your experience with our audience.
Great pleasure.
Thank you. Perhaps one day we will have the pleasure of welcoming you here in Brazil for an in-person visit. Right?
That would be nice.
Thank you once again for being.
Okay. Enjoy the rest of the conference, everyone.
Thank you.
Thank you.
Okay, for the second talk of our event, please join us using the new YouTube link that was sent to you. Thank you, everyone.